So now in section 13.2, we're going to talk about the regulation of transcription in bacteria. Now remember, there are two types of genes. There's constitutive genes whose expression is not regulated, who are constantly being expressed. However, there are other subsets of genes that are only expressed when required. And in order to initiate that process of gene expression, we use proteins called transcription factors, which are accessory proteins that are going to aid the enzyme RNA polymerase in initiating transcription. How do they do this? Well, that promoter has regulatory sequences within the DNA that allow these transcription factors to bind. And we're going to see when transcription factors are bound in the vicinity of the promoter, they have the ability to either increase the rate of binding by RNA polymerase, a constant we're going to call Kb, or they're going to affect the rate of isomerization, which is that open complex, which is the rate-committed step within transcription. So there are two types of transcription factors. There are ones called repressors. Repressors are going to exhibit negative control. They are going to downregulate transcription and its rate. So how can they do that? Well, they can make RNA polymerase bind worse to its promoter by simply binding in the same place RNA polymerase would have bound. Or they can act on the rate constant, inhibiting the formation of open complex, a lot like an enzyme inhibitor. On the flip side, there are proteins called activators which increase the rate of transcription. How do they do that? By increasing the rate of binding, making RNA polymerase bind better, or by increasing the rate of open complex. So here we go. Let's establish ourselves. Remember, a structural gene encodes for a product like protein. Flanking that upstream, as we've talked about previously, are promoter sequences within the DNA whose function it is to bind RNA polymerase and really establish the 5' end of the gene. Well, we can see at this position when this repressor binds, even if RNA polymerase is able to recognize its promoter, it's not able to then read the structural gene. It's occluded by the presence of this bulky repressor. Here we can see activator proteins binding in and around the promoter sequence actually are going to interact and recruit RNA polymerase, enhancing binding, and or put RNA polymerase in a conformation that allows it easily to open up the DNA templates and allow mRNA transcription to occur. So those are two approaches that a bacteria can take in order to regulate gene expression. Now, in addition to proteins, like repressors and activators, small molecules can also act as signals in order to regulate the transcription factors. When these transcription factors bind to these small molecules, we know they change conformation, and we know that conformational change can ultimately impact the functionality of that protein. Now, when you look at these transcription factors, we know that proteins have domains that can function independently of one another within an individual peptide chain. There are two domains that are going to be very important in the function of these transcription factors. The first is the effector binding site, which is in charge of binding that small molecule, which ultimately regulates the activity of that protein. And there's also a DNA binding site, which has to recognize specific promoters. And I think this is extremely important. There's not one transcription factor for every gene in bacteria. There are transcription factors that regulate specific genes. And therefore, it's very important that we demonstrate specificity between the promoter DNA and the DNA binding site within the protein. So here's a great example of how a small effector molecule can impact the repressor protein. So let's say there's no transcription of the gene currently. Let's say this is a protein that's involved in lactose metabolism. And when there's no lactose around, why express the gene? So this repressor effectively shuts off expression of that structural gene. Now let's say lactose is available. Well, imagine this scenario. The lactose binds a repressor protein, causing it to change shape. In this conformation, there is no DNA binding. Well, if the repressor can't bind the promoter, now RNA polymerase is able to effectively and efficiently express the genes to make lactose proteins. So lactose itself is a small molecule that then turned on expression of the genes involved in metabolizing itself. So it's a very elegant way, if you think about it, of a cell to conserve its resources and only turn on genes when the appropriate conditions are met. And it's these small effector molecules that allow the cell to identify those changes. So how do we organize genes in a prokaryotic cell? Well, we organize them into operons, and this is unique to prokaryotes, so we have to establish that. An operon represents a cluster of genes with similar functions under control of a single regulatory region called an operator. So we can have gene A, B, C, and D, 4 independent genes within the DNA that all contribute to a common function. Well, listen, as a bacterial cell, why not group these together? And that way, a single regulatory event at its promoter can express all 4 genes at the same time. That helps a bacteria coordinate and strictly regulate its cellular processes. So how can it do that? Well, when it transcribes these genes, it transcribes them into a single mRNA called polycistronic RNA. So here's the RNA that's produced. Here's a 5' end, 3' end. There's a start codon here for gene A, a start codon for gene B, a start codon for C, and then you can imagine D here as well. And so one mRNA contains coding regions for 4 different proteins. So again, in a single step, in a single transcription step, and translation of a single mRNA, we get 4 independent proteins that all serve a common function. So we're going to talk about then different ways in which bacteria can regulate expression of the operons. We're going to see how they can induce it, meaning they turn it on, or we can repress it and turn it off. The lac operon is sort of the classic example that's been well studied in E. coli. The function of this operon is there are a set of genes that are involved in lactose metabolism. So I've tried to use this example previously, even though I didn't call it the lac operon at the time, to start to help frame the discussion here. And so when we look at this operon, there's a regulatory region, right, the LAC-P promoter, followed by then 3 structural genes. First is a LAC-Z gene, which encodes for beta-galactosidase. Beta-galactosidase is an enzyme that hydrolyzes lactose. There's also now a second gene, a permease gene, lactose permease, whose function is to transport lactose into the cell. The last gene is LAC-A. It's a galactosidase transacetylase. Essentially, it adds in a 2-carbon acetyl group to the molecule. Now, there are 2 regulatory sites near the promoter where RNA polymerase would bind. There's a LAC-O site, which binds the repressor protein. There's also a second site called a CAP site, which binds an activator protein. So we're going to see the lac operon can be both repressed by a repressor protein and activated by an activator protein. It's very important now. We draw the line here. So that's in the immediate region. You have a promoter, you have an operator, and then you have the lac C, the lac Y, and the lac A genes. Right? So that's sort of one unit. Now at a distant site, well away from that operon, is another gene called lac I. It's a structural gene that encodes for the lac repressor. Now, it's considered a regulatory gene, right? We have metabolic genes, we have genes involved in expression. Here we have a gene that's regulatory in nature, meaning its only function is to regulate expression of another set of genes. It has its own promoter, it's regulated in its own set of events. It is not regulated by lac O, lac P, or capsite. So that's very important to establish. And here's a picture, maybe a little bit better than what I drew, to orient you here. Here's the promoter with the operator capsite. Here's ZYA. Here's the terminator. Here's the corresponding mRNA. Notice it's polycistronic because it contains coding information for all three genes in a single mRNA transcript. So what are the functions of some of the products here? Well, the lac permease is a symporter using gradients of protons in order to bring lactose against its gradient, moving past equilibrium, allowing that bacteria to take in as much of this disaccharide as possible. Once it's in, beta-galactosidase is able to recognize this glycosidic linkage and able to cleave the disaccharide into its component monosaccharides, which is galactose and glucose. This enzyme also can carry out a second reaction, which can convert lactose to allolactose by simply changing the type of linkage that we see here. Rather than a 1,4, we can transition to a 1,6. That's not extremely important, the change in the linkage. But understanding that it can also convert lactose to a molecule called allolactose. Here's why that's important. Allolactose is the signal. What I mean by that, it's what's acting as the small effector molecule. So now, sometimes we like to bring poles in. Here would be an example. Let me So based on the lecture you viewed today, what would you consider an operon to be? You could go just to polev.com. Don't follow that address since this won't be live at the time, but go to polev.com and put in your answer choice on what best describes an operon. All right, so now let's look at the different scenarios which a cell faces, and under these conditions, how the lac operon is regulated. One of the things I want to establish now is that the enzymes involved here, if you think about it, are catabolic enzymes, right? Because they assist in the breakdown of these substrates. So it doesn't make sense for a cell to synthesize catabolic machinery if there is no substrate. It's only going to synthesize these components when the substrate is available to break down. So when lactose is absent, the lac operon is going to be in the off position. Now, how does that work? Well, it's quite simple. We know that there's a repressor protein from a distant gene, structural regulatory gene called lac I. When lac repressor is synthesized, in this case, it's active. It binds to the operator, preventing RNA polymerase from transcribing the gene. So I get no gene product when the repressor is bound at the operator. All right. So now, what happens when the environment shifts? What happens when the substrate is now available? I need to turn this on because I need to bring in the lactose and I need to metabolize it so the bacteria can get energy. Well, we still have expression of LAC-I. That has not changed. But what has changed is the activity of the repressor, because the production of allolactose by the few molecules of beta-galactosidase that are present in the cell is going to inhibit the repressor. It's going to change the DNA binding domain. It's going to change the conformation. No longer will the operator be occupied. And so now as RNA polymerase binds, it's able to move down the gene, creating a polycistronic mRNA that's ultimately translated into the 3 specific proteins. So how can we turn on this gene in this example? By downregulating or turning off the repressor using a small molecule, in this case a derivative of lactose. So there's no better way to know lactose is available in your environment than by using lactose as a small molecule, right? It makes a lot of sense. And so that's what allows the cell to transition from lac operon being off to the lac operator being on. All right, so which answer do you think best describes, which answer is best all right, so here's a question. Which of the following would occur if there were a mutation in the LAC-O site of the lac operon? And let's imagine this mutation would hinder or cause the binding of the repressor to be less specific. So it negatively impacts the binding constant. What would the best answer be? So what's the best answer for this question? Yeah, that's right. If the LAC-O site is mutated, it's the repressor that's going to be affected. And so a mutation in the lac operator is going to cause the operon to be in the default on state, because no longer will the repressor be able to efficiently bind. Second question, which of the following occurs in E. coli when lactose is absent? Yeah, that's right. It's going to negatively impact expression. I would take this stuff, black and white, makes it difficult at times. There is going to be some beta-galactosidase and permease present, as we established very early on in this chapter. A lot of that is from previous expression of the lac operon. But I think students have difficulty with this idea that there's none or trace, so I would have accepted B or C. And so really understanding of the function of the lac repressor was done using mutants of the lac operon. And so what they found is there was a series of mutants in which now the lac operon is constantly on. Now some of them ended up being in the lac-I region, right? And they couldn't make proper lac repressor. And so it's within understanding these mutations that really helped us establish the story. So the hypothesis was that by having a lac-I negative mutation, it resulted in the synthesis of an internal inducer that turned on the lac operon. So they originally thought that the lac-I mutation was encoding for a gene helped the production of a gene that induced expression. What they actually found out is that the mutation prevented the repressor from inhibiting the lac operon. And hopefully you can see those are two very different things. Rather than having another protein be able to bind and help transcription, it was simply a function of the repressor no longer being able to bind. So we just talked about the lac operon being under negative control of a repressor. That repressor's activities only alleviated when lactose was available. But there's more than just lactose that's in charge of regulating expression of this operon. See, glucose levels also impact the expression of the lac operon. Now, why is that? Well, if I'm a bacteria and I have two available substrates for metabolism, glucose and lactose, which of those is the preferred substrate to run glycolysis and subsequent reactions? Yeah, that's right, glucose is. So if both glucose and lactose are available, I'm going to prefer glucose. So I don't want to metabolize that lactose. It's only when glucose becomes depleted will I need the lac operon. So here's another set of conditions in which the cell needs to be able to react. And it does it through a repressor, it does this through an activator protein called the catabolite activator protein, otherwise known as CAP. See, there's catabolite repression that occurs here because as we said, when glucose is available, we don't want the lac operon to be on. So glucose glucose-rich conditions function to repress the lac operon, but not through glucose itself. It's through a small effector molecule called cyclic AMP, the same cyclic AMP we've seen in cell communication. It turns out that there's an inverse relationship. So as a cell encounters high levels of glucose, we are going to have low levels of cyclic AMP. So glucose represses the cyclic AMP levels. However, when glucose is at low concentrations in the cell, cyclic AMP levels subsequently rise. So it's inversely proportional. Now, why is this important? Well, the reason that this is important is this activator protein called CAP requires cyclic AMP in order to be active. So CAP by itself, which turns on the lac operon, is inactive. When CAP is bound to cyclic AMP, it's now active. And when it's active, it can turn on expression of the lac operon. This is what we call positive control. Just as a repressor showed negative control because it turned off gene expression, CAP regulation is an example of positive control for this operon because it upregulates gene expression. And it does so by binding a unique site just upstream of the promoter. When CAP is bound at this site, it's able to make productive interactions with RNA polymerase to help it bind, in most cases, although in some cases, it's also going to help establish that open complex. Once RNA polymerase opens up the DNA templates, RNA production must occur. Transcription is off to the races. And so in each case, the rate of transcription is increased by enhancing binding and enhancing the rate of isomerization. But how does this happen? How do we transition from active to inactive? Well, when glucose is present, CAP is inactive, meaning the lac operon is not turned on. When glucose now levels drop, it's going to turn on CAP in order to induce the expression of lactose when it's available. That's an extremely important point. If there's no lactose here to be had, the repressor is still going to be bound at the operator. But now if lactose is available, we know repressor isn't a concern. And when glucose isn't available, we know CAP can now bind. And so we can see two very different modes of regulation of the same operon based on the conditions which the cell faces. So now here's a summary. When both lactose and glucose is high, the operon is shut off. Why? Because CAMP is low. When cyclic AMP levels are low, CAP is in its inactive state. When it's inactive, it cannot induce expression of the operon. But when lactose is high and glucose is low, now the operon's turned on for a few reasons. First is the repressor is not in its active state. It's inactive due to the presence of allolactose. And because there's no glucose, there's enough cyclic AMP to activate CAP. And when CAP binds, it enhances RNA polymerase activity to make and express more of those lac proteins. It's a really elegant system. It's establishing what's happening in an environment, and it's making changes in gene expression in order to respond. Now, when lactose is low and glucose is high or low, the operon is shut off. The reason being, no lactose, no allolactose, the lac repressor wins out. No matter if CAP is bound or not, the repressor will not allow RNA polymerase to establish itself and to express the operon. Now here's a nice video that's available for you to view on our website, on our course materials. I strongly suggest you go check it out so you can really see it in 3 dimensions and see how all these proteins are working together to regulate expression of the operon. So now let's answer this question. Let's break it up. The ability of glucose to repress the lac operon depends on which of the following effector molecules? That's right, cyclic AMP. Because without cyclic AMP present, CAP is inactive. And when CAP is inactive, the lac operon is off. Here's another example of an operon arrangement. Now, this is a little bit different example, and I want to highlight the differences now. There's a set of genes that are part of what's called a trip operon. Tryptophan is an amino acid that is essential for translation, right? It's one of the 20 amino acids. Well, it turns out that E. coli has the enzymes required to make tryptophan, meaning that these are enzymes that are anabolic in nature. Whereas the lac operon produced enzymes that were catabolic in nature. So this is going to be very different, right? Because for catabolic enzymes, I don't want to express them until the substrate is present. Until lactose is available, I'm not going to turn on the lac operon. Here's the opposite. I need this operon to be on when tryptophan is not present in the environment because I need these enzymes to make it. However, when tryptophan is available in the environment, why continue to waste energy making anabolic machinery? So when tryptophan is present, I want to turn off the trip operon and rely on the existing substrates available to help make protein. Remember, gene regulation is a lot about conserving resources. If I have a resource available, why continue to make it myself and waste precious resources? And so what is the signal or the small effector molecule that allows the operon to be regulated? It's tryptophan itself. The tryptophan is going to act as a repressor molecule. So when it's available, it's going to act as a co-repressor, turning off expression. Let's take a look at it. There are 5 structural genes, all of which contribute to tryptophan biosynthesis, produced as a polycistronic message. When tryptophan is low or absent, RNA polymerase is going to freely bind and express it. So the default state here is on. Now let's imagine that the bacterium encounters tryptophan. Now what happens? Well, just as we saw before with lac-I, there's a distant regulatory gene called TRPR, whose gene product is a repressor which binds to its operator to repress transcription under very specific conditions. So even when tryptophan is absent, the repressor is produced but not active. It's not in its active conformation. However, now, when tryptophan is imported into the cytoplasm, the tryptophan can bind the repressor, converting it to its active conformation. And in its active conformation, it can bind its operator, turning expression of these genes off. So now, lac produces catabolic machinery. Its default state is off when no substrate present. Because why make the enzymes to break down lactose if there's no lactose to be had? Tryptophan is part of an anabolic pathway. It is on when no substrate is present. Because I need to make tryptophan. However, when tryptophan is now available, I'm going to shut the pathway off by using tryptophan as a way to regulate the activity of the repressor. And that's essentially what this slide says. The lac operon is inducible. I can turn on expression, and I want to turn it on in response to substrate availability. On the flip side, the TRP operon is repressible. It's off in its it's on in its default state, and it's only turned off when substrate now is present.